Hello, and welcome to this week's Ethical Dilemma in Science, where we're going to take a look at Guinea worm disease and ask the questions: Are we looking at eradicating an infectious parasite, or are we undergoing the targeted extinction of yet another species? So, taking a look at this, again, trying to pull this into current events. So, we've got an article here from the journal Nature from earlier this month, February, look at the date, February 11th, 2022, indicating that the worldwide incidence of Guinea worm disease is estimated at 14 cases in the year 2021. So what's going on? What is Guinea worm disease, and why is it being eradicated? What's the issue here? And so basically what we can see over here on the right-hand portion of the slide is taking a look at the number of cases over the last 45 years, going back, I guess maybe 35 years, doing the math in my head, from about 1985 upwards to about 2020. And this graph is showing it kind of dropping off here. Look at this. This is a log scale, so it's a little bit misleading. So this is 1 to 10, 10 to 100, 100 to 1,000, 10,000, 100,000, million, 10 million up here. So we got a huge drop that's occurring, but it's even greater when we look at the magnitude along the y-axis here. And so basically we're seeing a situation where about, in the 1980s, about 35 years ago now, if we take a look at this, we had about 3.5 million infections of this parasitic worm in 20 tropical countries a year. And with no recognized treatment or vaccine, there was an effort made to eradicate this, to prevent the transmission of this disease out there in the wild. And so the Carter Center, established in the 1980s, foundation established by former U.S. President Jimmy Carter, has helped lead this effort in the eradication of the Guinea worm since about 1984. So basically, what is it to kind of eliminate a disease? And this is kind of novel because if we take a look at this, at the history of humanity, we've only been able to eradicate 2 diseases in our entire history. The first is smallpox. So over here on the left-hand portion of the slide, we've got an individual here with the traditional smallpox blisters being present on the arm. And this is associated with a fairly severe series of epidemics throughout the history of the world. It's even estimated that between 300 and 500 million people died of smallpox in the 20th century. This corresponds to about 10% of the deaths, even with 2 world wars, the Spanish flu epidemic, all these other things that have been going on. About 10% of the deaths were due to smallpox. The other disease that has been purposely eradicated, is rinderpest over here. So we've got a photograph here showing a herd of cattle. It affects cattle and buffalo, and basically causes the death. It basically wipes out cattle herds pretty dramatically. And it was very extensively within Europe and Africa in the 18th and 20th century. And so there was an intentional effort to kind of eliminate the transmission of this disease through the process of vaccination and basically preventing infected individuals from spreading it to other individuals. Now, even though we're talking about this as eradicated, and we'll come back to talk about the definitions of eradication later on, we still have samples that are stored of both the smallpox virus and the rinderpest virus. And so it's been eliminated from the wild and kind of natural transmission in the wild, but there are stocks reserved for future research. Within smallpox, we're looking at kind of samples that are stored within designated research facilities by the World Health Organization. They've actually got an advisory board, the WHO, the World Health Organization Advisory Committee on Variola virus research. And there are 2 designated sites in the world where these smallpox viruses are stored and researched. There's one within the United States and one within Russia. In terms of rinderpest, there were viruses stored in a number of locations for research. There's now an effort out to sequence the genome of the rinderpest virus and then ultimately eliminate the stocks. But currently, there are still stocks maintained within Ethiopia, Japan, the United States, and China. And so, going back to the guinea worm, what is the guinea worm? Basically, it's going to be a parasitic worm. It's going to be found in kind of stagnant pools in locations within the tropical regions. And so the larvae, basically the baby worms, get within these little, roughly the equivalent of little fleas that are present within the water. Individuals may drink that contaminated water, and in doing so, they will ingest these larvae. Now, the digestive system, your body will attack and kill the fleas, but the larvae are going to be able to grow. They're going to grow, they're going to reproduce, and basically, for an individual that's been infected with this parasitic worm, it can grow within their body for anywhere to, a year, maybe 14 months at the outset. But at that time, basically, as it's feeding, as it's growing, the females are going to be producing lots and lots of eggs. Those eggs are going to develop into, basically baby worms, larval worms. And then about a year after that exposure, a year after the start of the infection, that large female worm is going to burrow through the skin, and usually kind of in the area of the feet or the lower legs. They're going to form a blister, and that's going to be a very painful, very burning blister. And so people that have these blisters that are forming are often going to try to, kind of cool off or soothe the pain that's coming. And so they'll go and soak their feet in water. But in doing so, what's going to happen is that this guinea worm is going to start to emerge. And as it emerges and comes into contact with water, it's going to release those babies. It's going to release those larval worms that are going to, again, get into the water and can cause further transmission of this disease. And so there are a couple of things that are going to be going on. If we take a look at this, it's a very characteristic appearance of these guinea worms. And so this is a worm that has been in this individual's body for upwards to a year. It formed a blister, and then when that blister burst, the worm is going to start to emerge. And so it's going to start to emerge fairly slowly. And so these worms can grow upwards to about 3 feet in length. And so basically a simple measuring tape here. Take a look at this. This would be kind of emerging from the foot. And then over time, it's going to get a little bit longer as it merges further and merges further. And we end up with this situation that whenever this comes into contact, it is going to release these larvae, or come into contact with water. It's going to release the larvae in the water and spread that. And with this growing up to, potentially, 30 inches, going through here, that's about a foot, about 2 feet. So we're talking about a worm, that it's kind of this long. And if it breaks off, the worm is going to die. If parts of it are within the body, it can contribute to the kind of rotting of the worm and the risk of a bacterial infection. So individuals that have this guinea worm emerging will often take it as it's emerging from the skin, start to roll it up around a stick. I got a pencil over here, a pen, I guess I grabbed a pen. And so as it's emerging, they kind of roll it up and kind of keep control of that. And again, prevent it from, breaking off and dying, and allow you to completely remove the entire worm from the individual. But again, that's a situation where this worm then, if this individual comes into contact with water, can potentially spread these baby worms, these parasites, to the water where they can infect other people. And so the Carter Center, leading the effort since about 1986, working with a number of ministries of health and local communities, the United States Center for Disease Control and Prevention, the World Health Organization, UNICEF, a variety of other areas, have focused in trying to prevent the spread of this disease. And the way they're going to do that is to prevent both the kind of ingestion of these parasites, the worm larvae, the baby worms, from a contaminated water source, as well as kind of preventing infectious people over here, Guinea Worm Containment Center, taking a look at keeping these infectious people away from where they contaminate water. And so prevent them from going into water for bathing or for cooling it off. This does affect animals such as dogs, so it affects a dog, keeping the dog out of the water can't prevent that worm that's emerging from the body from establishing that next generation of contaminated parasites or parasites within the water, contaminating the water. There are often efforts to, when they have a guinea worm infestation, to treat the water with pesticides to try to kill that off. And then they're also, encouraging people to kind of avoid eating or letting dogs eat raw fish, fish entrails, things like that, to reduce the risk of taking up these parasites. They've also established these filtered drinking tubes, basically a straw with a filter across from it, so that people can drink from a water source, and the filter prevents the larvae within those water fleas from getting into their body. When they're collecting water from sources, they're going to have like cloth filters that'll put across the container that they're pouring the water into to, again, prevent the spread of this disease. And so this gives rise to some of the ethical dilemma and science questions that we can ask for this topic. Should a reference sample of, I'm going to pronounce this very poorly, Dracunculus medinensis be maintained similar to smallpox? And so the Dracunculus is the virus that causes Guinea worm disease. Now, as I said earlier, we've eradicated 2 diseases through intentional efforts of humanity, smallpox and rinderpest. But smallpox, even though it's been eliminated from the wild, is still present within the research setting. There are samples that are available for research, and you watch bad adventure movies, the risk that they will be, kind of stolen and released onto the world. And so this gives rise to kind of several definitions for eliminating the spread of this disease, to eliminate the transmission of this disease. The first definition is going to be this one of elimination. Elimination is basically stopping the transmission of the disease within a limited geographic area. So going in a very intentional manner, kind of prevent the spread of the disease, prevent potentially host vectors that would be carrying the disease for a period of time from getting infected, and block that. Now, in areas where the disease has been eliminated, you still have to have ongoing control mechanisms to prevent retransmission or new transmission of the disease. And so it's a very early kind of preliminary stage at eliminating the transmission of the disease or stopping the transmission of the disease. Eradication, again, is a step higher than that. Eradication would be the kind of intentional removal of the pathogen, in this case, the parasitic worm, to stop the transmission. And so if you've eliminated it worldwide, like we've done with smallpox, you don't need ongoing control mechanisms. And so there's no need for ongoing smallpox vaccinations because there is not smallpox virus circulating around the world. Now, with Dracunculus, this parasitic worm, we don't have viruses or vaccines won't work on it to eliminate it, so we got to come up with other ways, and that's why we're focusing in on eliminating the transmission of these parasitic worms, preventing them from getting into the water supply, contaminating it, and then getting into new individuals and infecting those individuals, keeping the life cycle going on. Now, even though we've eliminated smallpox and rinderpest from the wild, we still have it in the samples. And so until we can basically eliminate them from both nature and the laboratory setting, we haven't caused the extinction. And so the question is, should we be looking at the extinction of this parasitic worm? Should we be maintaining, samples of this worm, in a secure location? Similar to what we're doing with smallpox for potential future research later on. So that's the first ethical dilemma in science. The second one, a little bit more philosophically, should a living species be targeted for eradication? If we take a look at the history of mankind and the history and impact of humanity on the environment, kind of historically and ongoing, we can see that, intentionally or unintentionally, we've caused the extinction of many different species. Now, if we want to take a look at kind of preventing a dangerous or difficult or painful disease from being kind of spread throughout the community, we can take a look at eliminating a virus like smallpox. And by eliminating the virus, by the definition that biologists use, a virus is non-living. It, has biological molecules associated with it. It is capable of taking over an individual's, a living organism's cells to create new virus particles, but it really isn't living on itself. And so we can target a virus like smallpox. We can target a virus like rinderpest. But should we consider, the impact of the targeted extinction or eradication of a pathogenic species? Should we intentionally be eliminating one of these species, a living species? In addition to that, a related question: should potential beneficial interactions with other species be considered when targeting the disease? Now, in the case of the guinea worm, it's pretty straightforward. It does not appear to have any positive or beneficial relationships with other organisms or other species. So therefore, it becomes easy that, if it doesn't have a beneficial effect with, any organism that it interacts with, maybe eliminating from this from the environment, eliminating it from, existence, isn't going to have a ripple effect on the ecosystem. But what should be doing? What should we be considering if we're taking a look at eliminating or eradicating an organism that has potentially beneficial interactions with other species? It may be, harmful to humans. It may be harmful to, maybe we're looking at an agricultural pest of some type. We want to eliminate, crop loss. But it may have a beneficial effect on other organisms. Where is that balance on determining whether it's beneficial to us, harmful to us, or beneficial or harmful to other organisms? And so where should that line be for determining or targeting organisms for eradication or extinction that could have potentially beneficial interactions with other species or could have an effect on the ecosystem if they were eliminated? I'll post these references below in the links within the description, so a variety of both popular press articles as well as some additional more scientifically oriented articles as well. As always, if you have any questions, please reach out to me. I'll put these links in the information below. Thank you, and please come back for next week's Ethical Dilemma in Science. Thank you, and have a great week.